Morning, everyone. I'm Robert Reed. I'm Director of the Biomedical Research Centre at University Hospital Southampton, University of Southampton. And I'm also Saul Forst, who is the Director of the NIHR Southampton Clinical Research Facility. Saul, my alter ego, is currently pinned down by the BBC, getting all the credit for the booster study that has basically shown that the RNA vaccines are very effective booster vaccines for COVID-19, and that's a fantastic piece of work which was led out of Southampton and of which we should all be very proud. So well done, Saul. Excellent. So he can't be here, so I'm the ability to tell you about the NIHR and clinical experimental medicine and clinical translation in Southampton and what's available to you. Well, this is basically a map of the NIHR, and it is a huge organisation. Essentially, it's about 15 years ago, it absorbed all of the NHS funding resources, and Sally Davis actually basically put it into this kind of shape. So the NIHR funds primary research, research grants. It's always human research. It's never animal research. And it has also funded trainees. So in Southampton we've got many trainees, clinical trainees, healthcare professional trainees who are doing PhDs, who are doing postdoctoral research. It's always, though, on human disease. There are data science facilities, and over here on the left is the infrastructure that they fund. And this amounts to about 2 billion of infrastructure, just in terms of the biomedical research centres and the clinical research facilities. So they fund centres in specialist units that are capable of doing early phase to late phase clinical research, and also they fund centres that do experimental medicine research directed at human disease. And the reason I'm telling you about this is because in Southampton we have these resources available to you. So the first of these is that I would like to mention to you is the Clinical Research Facility. And this is a facility that is in the middle of the University Hospital Southampton. UHS is a huge hospital. It's unusual amongst UK cities, and it's actually the only major hospital for some distance, and has a massive caseload. And of course, it serves a large population with high deprivation indices, and it's big. I mean, compared to other CRFs up and down the country, it's a huge clinical research facility. It's been there for 15 years now. It's developed. It's getting even bigger. It has got inpatient. You can actually do experiments on patients as inpatients. There's a large outpatient facility. There are negative pressure suites where you can do invasive procedures involving the airway, for example. And there are all sorts of physiological opportunities and imaging facilities that enable you to do experimental research. Linked to the CRF, there are specialist laboratories. These include the one shown here. There's a mass spec lab. There's an AMR lab that I will come on and talk to you about in a few minutes' time. But also the CRF is linked to the Wessex, the WISH lab, the Wessex integrated. I can't remember what the acronym stands for, but it's the WISH lab, which is an MHRA accredited laboratory facilities that can do measurements that can be used in clinical trials of investigational products. So it's that kind of grade of facility that is available. The CRF, as I said, is right in the middle of the hospital, and the reason why this is important, literally the intensive care unit is upstairs. It's surrounded by relevant wards, and it is such an easy facility to use. So you can base your clinical teams within the CRF, within the office facilities there, which are have excellent facilities for everything you need to help you conduct your research, and yet those individuals then can easily traffic to their clinical departments to do their clinical work or get clinical samples. Now, for those of you who don't work in a clinical environment, why is this relevant to you? Well, it's very likely that you're making discoveries that are relevant to clinical medicine. And for you, it's probably an obscure challenge. How on earth do you get to translate your discovery into something where there is an experimental medicine output? Well, it depends on how far along that pathway you are. But I mean, if you're early in the pathway, you can come to what the Biomedical Research Centre, which I'll talk to you about in a minute. But if you're late in the pathway, if you're very close to translating, then the CRF facility has staff who can help you construct a protocol to do a research in humans and help you get it through the ethics and the other governance instruments that are necessary for you to translate your discovery into a human medicine or an early experimental medicine study. So it is relevant to you, because if you want to do something in humans, you can come to us, either to the CRF or, if it's earlier on, to the Biomedical Research Centre, which is here. Sorry, you can see that this is a draft, but this is what the BRC actually looks like, and I'll explain. We were funded in 2017 to have an evolved biomedical research centre funded by the NIHR. The infrastructure award was about 15 million. The research was focused on nutrition and respiratory and critical care, with cross-cutting themes of microbial science, data science and behavioural science. And each of those elements were funded to a greater or lesser extent. Microbial science was around about 200k per annum, but it was essentially just to try and support the main themes of research, which included respiratory research and nutrition. And as things have evolved over the last 5 years, we are now moving towards a reapplication for the BRC. We've been shortlisted, and we'll find out in May if we're able to proceed. But the plan is that we will have 6 major themes, which will all be funded to the tune over 5 years of approximately 3 million each, with core infrastructure funding as well. And the themes are going to be respiratory and allergy, data health and society, so using bioinformatics. Respiratory and allergy will be led by Tom Wilkinson, Graham Roberts. Data health and society by Wendy Hall and Annika Lucassen. Microbiology, immunology and infection, relevant to this group today, will be led by myself and Saul Forst. Perioperative and critical care will be led by Mike Grocott and colleagues. And supporting behavioural change will be Lucy Yardley and Mary Barker. And finally, nutrition, lifestyle and metabolism by Keith Godfrey and Jonathan Swan. Now, these two here are the current major themes, but as you can see, we've expanded the major themes that the Southampton BRC will have, provided we get through the process. So, what do these themes do? They do experimental medicine. What does that mean? That means anything where you are trying to establish mechanisms in disease that are directed towards human health, anything that involves an intervention in particular. So in other words, not observational research unless you can specify an intervention and you're measuring gain or loss of function through that process. So that's what experimental medicine is. It's a rather loose definition, but essentially it's research on humans in a hospital environment. Okay, so what about our theme of microbiology, immunology, and infection? Well, this is what it looks like. The thrust of the theme is to prevent infectious disease and to augment infection control, to get to faster diagnosis, new treatments, and also, unusually for BRCs, we're going to go out into the community and we're going to do experimental medicine in general practice. So, which is an unusual thing for BRCs to propose to do. And so we're talking about, as we go along this menu, we're talking about early phase interventions, looking at new vaccines, looking at novel vaccines using human experimental challenge to address correlates of protection, early vaccine responses. We are investigating new materials for infection control. We are linking with informatics colleagues to understand the microbial genomes. We are looking at very novel diagnostic systems such as optoelectronic systems and other technologies for diagnosis. We're looking at applying point-of-care diagnostics. So many of you, some of you here will have been involved in developing potential point-of-care diagnostics. Well, Southampton's a really good place to look at early diagnostics because we've got a lot of patients and we are publishing rampantly. And in particular, Dr. Tristan Clark is developing a huge portfolio of research showing the impact of point-of-care rapid molecular diagnostics. We're linking with NBIC to look at biofilm disruption, and of course this is relevant to diseases such as cystic fibrosis, such as infected prosthetic joints, you know, we have the facilities available to look at clinical samples and the effect of molecules on the biofilm. And finally, we're going to take all of this out into the community and apply it where we can in general practice. A good example would be using rapid point-of-care molecular diagnostics to prevent antibiotic usage in general practice. So here are the assets as seen from our rather narrow perspective, but you can see that we have some very important connections throughout Wessex. We're also connected with other university hospital trusts in Wessex, not just with the University Hospital Southampton, it's quite a potent team sheet. We are, I think, a convincing group, and by that I mean everyone who does research in this region that has some connection with the BRC. We believe there's been over 600 publications just in the last 5 years, and there have been, just in terms of project funding, quite large grant capture. So going forward, these are our four research areas. Actually, you can see that practically everybody in this room can fit into one or more of these, but these are the themes going forward. And if we get funded, we are able to modify our objectives to fit the I mean, for example, 2 years ago we didn't know we were going to do COVID research, but we have been focusing quite a lot on that. Okay, so finally, I do want to mention that we've also acquired some infrastructure funding for antimicrobial resistance research, and this was a capital award that was given to Southampton, and the award was spent on making a lab that is literally it's one floor above the acute medical unit in University Hospital Southampton, and the award also equipped it with some useful kit. Our overarching strategy is development and testing of rapid and point-of-care diagnostics. And of course, the reason why this is relevant to AMR is because one of the big reasons for AMR, antimicrobial resistance, is the overuse of antibiotics. And that's all there is to it. If we could be more specific about therapy, we would be we would have less antimicrobial resistance. And also, of course, search for novel therapies in the widest sense. So, and in particular, biofilm disrupting therapies. So, this was built just as COVID was taking off, and so it's remarkable that it was built at all. But of course, with the restrictions within the hospital, it hasn't yet come into full use. But it's a nice facility. It's very close to the acute medical unit, as I've said to you. It's exceptionally good. It has seen action because it's where we were doing rapid point of care. Because it's so near to the AMU, we were able to do Tristan Clark's team was able to do rapid point of care using these modern multiplex PCR systems, which give doctors a fairly rapid idea of whether the patient has a virus infection that could cause their syndrome, and that allows you to move them down a pathway that doesn't involve antibiotics. And that's so important, and Tristan's team have demonstrated in a Lancet paper that it works in COVID. Here is the lab manager, Gavin, pretending to use a very good confocal in open lighting, but that's in the facility. But there are other very high state-of-the-art pieces of kit. We can't actually fit them in the AMR lab, so they are within the campus, and they're including stuff. I think Samit is going to talk about this at some stage, but it includes the use of CARs to do label-free analysis of clinical samples. So it would be possible to use this to measure the disruption of a biofilm in real time, and also things like Raman. Also, I think it's in the same place. This is in the, I believe it's in the IDS building. Samit will correct me if I'm wrong. But there are other pieces of kit. James Wilkinson and Nick Evans have acquired these unusual pieces of equipment that can do nanoanalysis, so looking at nanoparticles and how they penetrate biofilms and microbials and microbes. So that's it. I'm going to stop there because I'm bang on time.